My name's Conor McClenaghan. I have the honour of introducing our fifth EpIC seminar focusing on electrophysiology and ion channels. I'm delighted to introduce Andrew Glazer, who is our speaker for this month. Andrew is an assistant professor of medicine at Vanderbilt University. He's a geneticist, and Andrew's research from his undergraduate days in MIT through his PhD at UC Berkeley and then postdoctoral training with Dan Roden at Vanderbilt spans developmental biology, including stickleback evolution. So I think in 1 year, Andrew went from working on stickleback tooth evolution to ion channels. So that's an interesting journey. And most recently, his current work focuses on cardiac arrhythmias and ion channel genetics. So Andrew opened his lab at Vanderbilt in the summer of 2021. And in my opinion, he's driving some really exciting approaches at the interfaces of genetics and electrophysiology, and has done some fascinating work characterizing disease-associated mutations, which I think he'll tell us about today. So without any more from me, Andrew, I'll pass it over to you. Thanks for joining us.
Okay, well, thanks a lot, Connor. Yeah, it's and thanks to the rest of the organizers for the invitation. It's a really great opportunity, and I've really enjoyed the seminars so far, and hopefully I can live up to the high bar set by my predecessors. So today I'll tell you about some of the work that we're doing, studying doing high throughput studies of cardiac voltage gated ion channels. And I just wanted to start off by giving a little bit of motivation for why we want to do high throughput studies. A lot of you might be familiar with kind of lower throughput studies of one variant or a few variants and kind of really studying them in depth. So why would we even want to do these types of high throughput studies? A lot of the motivation actually comes from the clinic and from a clinical problem, which is basically that essentially the genetic testing is kind of taking off. And so lots and lots of people are getting their genome sequenced. We're discovering lots and lots of mutations in really interesting and important genes. And essentially, we're not keeping up on the functional side. We're not keeping up in terms of the rate at which we're studying the mutations. So here's a plot of variants in the ClinVar database of clinical classifications. And you can see that the number of variants over time that are missense is kind of going up like crazy. And this is a category of variants of uncertain significance, or VUS. And that's a category which basically means we're not sure whether or not it causes disease. And by comparison, the number of variants that are pathogenic or likely pathogenic, or benign or likely benign, is staying relatively constant. I mean, you can zoom in and you can see they're actually increasing slightly, but certainly they're getting way outpaced by the number of variants of uncertain significance, or VUS. And I should say this is across 15 major arrhythmia genes, most of which are ion channels. And so this is a clinical problem, which is my argument would be that we need to layer on top of the kind of detailed single variant studies. We need to layer a set of high throughput studies where we can study lots and lots of variants at scale to try to address this variant of uncertain significance problem. So how do we actually interpret a variant? There's these guidelines called the ACMG guidelines, American College of Medical Genetics, that were proposed and widely adopted since 2015. And this integrates together lots of different criteria about a variant. But kind of importantly for this talk, one of the major criteria that's used is in vitro functional data. So there's a real chance that if we actually study these variants in the lab and we get some results for whether or not they mess up the function of the protein or not, that data can actually be fed back into the classification criteria that are used when these variants are shown up in patients. And along the way, if we learn something about the biology of these genes and proteins as well, that's a big plus as well. Okay, so this is my one slide on the introduction to the cardiac action potential. The aficionados on the call will recognize this as a relatively simplified view, but essentially there's a series of cell membrane voltage-gated ion channels that pass sodium, potassium, and calcium ions in or out of the cell in a regulated manner. So kind of at the start of the action potential, sodium rushes into the cell, then a bunch of calcium comes into the cell and kind of prolongs the depolarization of the cell. And at the end, there's a series of potassium channels that kick in and kind of repolarize the cell. And if you have a variant in one of these channels which disrupts their function, you can get a Mendelian arrhythmia disorder. So it's basically a rare disorder where your heart is kind of at risk of suddenly stopping. So obviously we'd like to recognize which variants cause these disorders and kind of try to treat them. So a lot of the talk today, I'll talk about this gene called SCN5A, which encodes the major voltage-gated sodium channel in the heart. And so SCN5A encodes a protein NaV1.5, and we actually, there's a big variety of different arrhythmia disorders linked to it. The major two that we think about are loss of function variants, which are linked to a disorder called Brugada syndrome, and gain of function variants, which are linked to a different arrhythmia disorder called long QT syndrome. And, it's not quite as complicated as these simple arrows are shown. There's lots of other kind of risk factors for whether or not the patients will get disease, but essentially you can think about these either loss or gain of function variants in this protein as being important for arrhythmia risk. And like I said, this channel is really responsible, kicks in at the start of the action potential. It's responsible for most of the depolarization at the start of the action potential, and then it inactivates relatively quickly. So this is what patch clamping of SCN5A looks like. You get a rapid influx of sodium into the cell, followed by a rapid inactivation of the channel. And so from this, you can measure peak current, and some variants have reduced peak current, and that's been shown to be correlated with this risk of this loss of function disorder, Brugada syndrome. Likewise, we can zoom in on this kind of later part of the patch protocol, and most of the wild type channels inactivate to about 0.3% of the peak. So they're almost all inactive. There's a little bit of residual current. And then there's some variants that actually have a little extra current. So this is called late current. So it's about, this variant, for example, has about 2%, which we consider kind of a big amount of late current. And this is a small amount of current, but it's for a very long time. So it kind of adds up to a big effect. And that is actually a gain of function feature of the protein, which can be linked to long QT syndrome, which is this other gain of function disorder. So today I'll tell you about kind of two different stories. So one of them, they're both different high throughput assays of variant function. So one of them is using an automated patch clamp instrument. And then the second is using a different approach that uses sequencing, high throughput sequencing coupled to a multiplex assay, and that's called a demutational scan. So we've done a lot of work on automated patch clamp of SCN5A variants. This is an approach that was really spearheaded by Al George's lab at Northwestern, and we've kind of been some of the early adopters of it as well. We use an instrument called the SynchroPatch 384. This is a automated instrument that can patch clamp a 384 well plate of wells. You don't always get every well filled with a cell, but you get hundreds of cells worth of data in about a 30 to 40 minute experiment. And so you can run, we run typically for SCN5A, we run about 5 different protocols, kind of prodding the cell in different ways and measuring different parameters. The way we do this, we're doing these experiments in HEC293 cells. We generate stable lines that are expressing either wild type cells or different mutants. And then we do a series of different selection tricks to generate cells that are expressing about 90%, expressing a high level of our channel. And then we can get, on a good day, we get results that kind of look like this, where we get lots of cells worth of data. So our first study that we did for SCN5A variants was in 2020. And we studied about 80 different variants in SCN5A. I should say that the real benefit of this is you can study lots and lots of variants over and over in lots of replicate cells pretty quickly. So we studied about 80 variants in this first study. Some of the variants looked relatively similar to wild type. Some of the variants looked kind of partial loss of function, and some of the variants looked like total loss of function. And I should say we picked these variants because they actually showed up in patients with Brugada syndrome. So we suspected that they were loss of function. So you can see here, compared to some controls, which have all about nearly about 100% peak current density, we see some of the variants that were suspected to be Brugada syndrome associated also had kind of near normal function, but a good fraction of them, we found about 45 of them had either partial loss of function or basically dead channels, basically zero current through the channels. And so this really expanded our set of known Brugada syndrome associated variants. We did a little bit of structural modeling to try to kind of figure out why these variants were loss of function. So this is kind of a view if you were a sodium ion entering the cell, going through this little hole from the top. And so you can see there's some interesting kind of correlations and enrichments. For example, variants in this kind of central pore domain were more enriched for loss of function. We actually saw some variants in kind of simulations. This is the wild type residue here, which doesn't occlude the pore. And then this variant here actually is predicted to kind of stick into the pore and occlude the pore. Sure enough, it was loss of function in our study. There's some other kind of obvious stuff like this variant disrupted a disulfide bond and was loss of function. But kind of overall, I would say we found some kind of correlations, but you couldn't completely predict the in vitro function from the structural analyses. And so using this data set, we used the ACMG classification criteria at the time to reclassify variants. So you can see going into the study, there were a lot of this VUS, variant of uncertain significance category. And after the study, we could bump a lot of them kind of either up or down in severity, depending on the patch clamp results. So this is kind of the really cool application of this is that you can actually hopefully use this to affect clinical classifications. So one other idea is that we could potentially use this type of patch clamp data to study new variants that are seen for the first time in these large sequencing data sets. So there's lots of sequencing efforts going on. We participated in a study called the eMERGE study, which was a group of 10 academic medical centers across the US. And we did a project where we sequenced a lot of kind of your least favorite genes. These are genes that cause bad diseases in about 20,000 people. And we took any known pathogenic variants or likely pathogenic variants, and we returned those results back to the patients and physicians kind of as a genomic screening exercise. So could we actually do population genomic screening and find these disease-causing variants? And then we discovered a lot of VUSs as well. And so we used in vitro studies to help figure out which VUSs associate with the phenotype. And so this is the kind of data we get. So we get this is focusing on a few major arrhythmia genes, and each of these is a different variant. And you can see that there's a lot of VUSs, and a lot of them show up in one patient, maybe 2 patients or 3 patients if you're lucky. But you get a lot of very small sample sizes in terms of the patient data. That being said, there's some variants that showed up in a patient with a pretty serious disease. So we've color-coded this basically depending on the severity of the arrhythmia. And you can see there's some variants that I would call suspicious. So they're variants of uncertain significance, but they show up in a patient with a pretty serious arrhythmia. And so those were kind of the type of variants that we thought that in vitro testing could help kind of resolve. So we did automated patch clamp of 50 different VUSs that we detected in this study across a few different genes. So here's some of the data for SCN5A showing that some of the variants kind of show up as normal again. Some of them are actually gain of function in terms of peak current, and some of them are either partial loss of function or loss of function. This is kind of the summary of some of the SCN5A data. One of the interesting things is we get out a lot of kind of normal variants, which kind of makes sense that if you make just a bunch of random changes to a protein, you get a bunch of normal functioning variants. So it kind of depends on what your selection set is. If your selection set is a set of patients that showed up in a set of variants that showed up in patients with disease, then you get a higher rate of loss of function or gain of function. If your selection set is kind of the general population, you get a higher rate of normal variants. Here's a variant that actually caused this late current gain of function property. And then here's some data that Al George at Northwestern generated. And this is data for potassium channel KCNQ1. And so again, he found a bunch of their lab found a few loss of function variants, and they actually did dominant negative studies as well in heterozygotes, and showed that some of these variants act through a dominant negative mechanism, which makes sense because it's a tetrameric protein. Okay, so one of the things that we've been doing recently is actually going back and doing a lot of controls. And this sounds kind of weird, but basically there is this paper that got published a couple years ago, which basically said if you have a cool new assay, you can't just blindly apply it at a strong level in the classification scheme. You actually have to prove that it works. And the way that you prove that it works is you study lots of controls, lots of known benign or likely benign variants, like in the top here, or you study lots of known pathogenic or likely pathogenic control variants. So that's kind of what we've been doing. And we've actually been collaborating with a group in Sydney, the Ng and Vandenberg group, and they've actually been studying the exact same set of variants on their syncer patch. So kind of two independent labs halfway across the world, different cloning methods, slightly different analysis methods, et cetera. But we get essentially very similar results, which I think is really kind of validates to me that we're doing something kind of right. So you can see the Australia group made this plot because of how they spelled our institution. The other cool thing is that essentially most of the benign variants have relatively normal function, with one exception, this variant S216L, and almost all of the pathogenic variants have either partial loss of function or total loss of function in this assay. Again, with a couple exceptions. This one actually has big perturbations to other gating properties that make it loss of function that aren't shown in this plot. But this one actually looks like totally normal in our assay. So essentially we get kind of a mixed bag. So we get almost all of the control variants have the predicted properties, which is great. And that means we can implement the calibration scheme at the strong level. However, it's a little interesting that one variant from each category had kind of a discordant result. So we're currently undergoing some experiments to investigate those two variants kind of in further detail. And one possibility is we're doing all these experiments in hex cells. And so it's possible that the final behavior of this channel, which happens in a cardiomyocyte, isn't properly modeled completely fully in the hex cell model. So we're trying to CRISPR these mutants into induced pluripotent stem cell derived cardiomyocyte. But overall, I would this is kind of the type of work that in 2023 that we need to do to kind of prove that our assays actually do a good job and can be implemented in clinical classification. So just to zoom out for a second, there's actually, I would say, this field of kind of automated patch clamping study of arrhythmia-associated variants is kind of maturing. And, there's actually been, 6 or 8 papers over the past few years studying different genes and studying, dozens, in some cases, even hundreds of variants in these genes. So, I think it's a really exciting time to be doing this type of work. And there's lots more variants in the pipeline. So stay tuned. For some reason, all these papers seem to be published in even years. So I'm not super optimistic about 2023, but stay tuned for 2024 and some cool new patch clamping papers from that. Okay, so that's kind of the automated patch clamping work. And the real nice feature about that is we're actually able to get lots of different parameters that are pretty analogous to the type of parameters you would get from a traditional manual patch clamping study. Now I'm going to switch gears kind of completely and talk about a different type of work that we're doing, which is something called a deep mutational scan. And it actually generates a completely different type of data set. And the idea here is that we can actually make potentially nearly every possible variant in a gene and study it in a multiplex assay. And we're not studying these variants one at a time. We use kind of clever tricks to pool the mutants together and study them using Illumina sequencing. So this work was kind of, we didn't invent this idea. This work was, really pioneered by lots of others. And so this is an example kind of pioneering paper in the field studying the cancer gene BRCA1. And they actually studied, a few hotspot, the important hotspot regions of this gene and made nearly every single possible mutant and kind of have a map of every single possible variant and whether or not it disrupts the function of BRCA1. And if you're familiar with these curves, you know that usually this is the predictive value of how well this correlates with the known pathogenic and benign data. And usually, people go their entire careers without getting curves that are this accurate. So they basically get the right answer from their assays, and they have a predictive map going forward of every new possible variant that might show up in a patient. So a few years ago, we decided to try to port over some of these techniques to the ion channel world and to the arrhythmia world. And I should also mention that if you tuned into the first the first EpIC seminar, you heard Willow give a great introduction to his work on using this method to study ion channel and transporter genes. So we're not the only ones working on this type of work. But so kind of here's the idea of how we're doing these experiments. We have a gene on a plasmid that's wild type. We do kind of massive mutagenesis to introduce every single possible mutant into the gene. And then we have a special cell line that our collaborators made that it can integrate in, the plasmid can integrate into the cell line such that each cell is expressing a different mutant. And that's an important, we don't want one cell to express 10 different mutants because what does that even mean? We want each cell to express just a single mutant. And then so we have a pool of different hex cells growing in a dish that contain a pool of different mutants. And then we do some kind of assay. And I'll tell you more about the different types of assays we're doing. And so one assay is kind of a cell death assay where we kill off cells with functioning channels, and we're left with the ones with loss of function channels. So we're left with kind of a biased pool of channels. And then we use deep sequencing essentially to count the frequency of each mutant in the different pools. So maybe in the starting pool versus the final pool, what's the frequency of each variant? And we can infer kind of a score from those counts. And kind of the hope is that ultimately we can build a map of every possible variant in the gene and kind of which variants screw up the function of the protein. So we've been doing this experiment for a while now. Back in 2016, we were trying to teach ourselves how to do these methods. So we started off with mutagenesis of one codon. I should mention a lot of this work I started in my former postdoc lab, in the lab of Dan Roden, and kind of a lot of this work is kind of arisen through a close collaboration with his lab. And so this is an example of how we do the mutagenesis. We start with a mutagenic primer that has 3 ends in it. And so that actually encodes all 64 possible codons of that position. And then we do mutagenesis using this primer, and we actually introduce a pool of all 64 possible mutants at this position. And so this is kind of the pool that we get of mutants. We get a mixture of all mutants at that 3 base position. And so you can imagine expanding this out. So the first thing we did was we expanded it out to a 12 amino acid region. We made a library of nearly every possible single amino acid change in the 12 amino acid region, which included lots of missense mutants, which were ultimately the ones that we're interested in. But also as controls, it includes a lot of nonsense mutants, premature stop codons, as well as synonymous mutants, which we think should act like wild type. And the region we picked was so I should say something about the structure of this protein. This is SN5A is a really large protein. It's about 2016 amino acids. It has 4 major transmembrane domains that collectively fold into a kind of a complete donut, a complete channel through which the sodium flows. And there's 24 transmembrane helices, 6 in each domain. So we picked the S4 helix, the 4th helix of domain 4, kind of seemed as good of it as any of a place to start. So then the question was, what do we do once we have these pools of cells? How do we actually distinguish between the cells that have functional channels or not? So this is the assay that we came up with, which we call a triple drug assay. So the problem with the sodium channel is it really likes being off or inactive. It does not like being in the open state. So if you express SN5A in hex cells, the channel is just inactive. There's no sodium flowing through the channel. So what we do is we add 2 drugs which bind the channel and promote the open state. So that kind of biases it towards open state. Now you get a bunch of sodium flowing through the channels into the cell. However, the cells have a great way to deal with excess sodium in the cell, which is the sodium potassium ATPase. So they just take the sodium and pump it right back out, and they're totally fine. So then what we do is we add an inhibitor of the sodium potassium ATPase called Wabane, and now the cells are in trouble. So they have a lot of sodium rushing into the cell through the sodium channel, and the main way they have to get rid of it is gone. And so we think either that sodium overload itself or the compensatory ways that the cell's getting rid of the sodium is actually toxic to the cell and causes cell death. So we did a lot of optimization of this assay with different concentrations and treatments and timings and things. And we kind of hit upon a concentration of these 3 drugs, which seemed to work pretty well. And it seemed like it could actually distinguish between different classes of mutants. So, for example, here's cells expressing wild type SCN5A. And you can see that I'm not going through the entire details of how we're measuring this, but essentially lower on this graph means that more of the cells are dying compared to controls. So we're getting a decent amount of cell death for cells expressing wild type channels. Cells expressing, and this is kind of an allelic series of different types of SCN5A mutations, ranging from low function to normal to high function. And so a stop codon, W822X, that actually has near 100% compared to controls. So that has very little cell death in this assay. This is a partial loss of function that has a little bit of sodium coming through the channel, and that was kind of an intermediate amount of cell death. This is a mutant that in patch clamp studies behaves pretty much like wild type, and that one had a result in this assay similar to wild type. And then this is a variant that actually has a gain of function property that's linked to this long QT syndrome. It has this late current feature. And this variant actually, kind of to our surprise, it actually died faster than wild type. So there's more cell death in this assay. We weren't really expecting this, but it kind of indicated to us that maybe this assay could be sensitive to detect both loss and gain of function variants, in that the loss function variants would die less fast compared to wild type, and the gain of function variants might die faster than wild type, or more than wild type in this assay. So we did the full experiment. We took our pool of all the mutants, we put them in the cells, we threw on these 3 drugs, and then we sequenced the initial pool and we sequenced the final pool. And we did a little bit of math. Wasn't super complicated, but we took the ratio of the abundance in the final pool divided by the initial pool. We did some scaling adjustments, and we get these data sets. So essentially what we do first is we look at the nonsense and the synonymous variants. And you can see here that there's 12 possible stop codons in this 12 amino acid region. There's 12 points here that are all kind of, there's some noise, but they're all normalized to be around 0. The synonymous variants are normalized to be around 100%. There's 12 of those as well. And then the missense variants look like this. So there's kind of a big spread. This is about 240 different missense variants. And you can see some of them have relatively normal function, kind of similar to wild type, relatively normal scores, let's say. And then some of them appear to have either loss of function scores or gain of function scores. And this corresponds to kind of dying more or less. These ones are dying more in our assay, and these ones are dying less in our assay. So we were kind of interested to see if we got the right answer. So one of the things we did is we pulled out a few of these variants to patch clamp and see if, and these were novel variants that had never been seen before. And those 3 variants are shown here, and essentially we got the right answer for all 3 of them. I won't show you all the data for that. And then we also compared, one of the reasons we picked this region of 12 amino acids was that there were a few known variants that had been already seen before and already either been studied by patch clamp and also seen in patients with disease. So we compared our data sets, our DMS scores, to those previous data. And kind of overall, the result was that out of the 9 kind of control variants in total, we get the quote unquote right answer for 8 of the 9. And so that's pretty good. We're pretty happy with that. The one that we actually had a discordant result for was actually really interesting. So this variant, R1626H, had a relatively normal, relatively wild type like DMS score. And on patch clamp, it has kind of a very complicated mixture of different gain and loss of function phenotypes, which, depending on which parameter you look at. And kind of my theory is that in our experiment, those kind of get all averaged out to kind of be like a wild type function level of current. So I think it kind of illustrates that we're never going to, we're taking a complex 2,000 amino acid protein and collapsing the behavior down to a single number. So we're never going to be able to be capturing all the complicated features that you would get by an actual patch clamp experiment. But still, I think you can get kind of trends and overall, certainly, the kind of the big loss of function or gain of function variants we think we can capture with this assay. Okay, so this was the heat map we get. It's kind of a puny heat map by deep mutational scan standards, but essentially we have different positions in the protein, different amino acids, and kind of a map of which mutants cause normal function or loss of function or gain of function. So then the next question was trying to scale this up. So and trying to scale it up by 168-fold, which was the size of the, scaling up from 12 amino acids up to 2,000 amino acids. And so the first thing we decided to do was kind of cheat and only study half the protein. And so if we looked at actually where in the protein are the disease mutants, and they're actually not evenly clustered throughout the protein, they're kind of clustered in certain domains, and those domains correspond to the transmembrane domains of the protein. So for example, here's the variants that show up in patients with Brugada syndrome, which is the loss of function disorder. And you can see that those variants are enriched in the kind of the structured domains, especially the kind of the latter half of the domains, which are actually the pore forming units, compared to the flexible intracellular linkers, which are largely stripped of largely depleted of Brugada syndrome mutants. So we decided, okay, let's just study the 4 major domains. We're not doing the exact domain boundaries, but kind of about half the protein that approximately corresponds to the 4 major domain boundaries. And so this is some of our data. So this is still a work in progress. We're kind of currently working through, making all the different libraries and throwing the drugs on them and sequencing and things like that. And there's kind of a big team of people kind of trying to carry out this experiment. But we have some data that's kind of hot off the presses for you guys for domain 3. So this is I kind of wish we had designed it a little differently, where we grabbed the first and second helices as well, but this is actually helix 3 through 6 in the transmembrane in domain 3, as well as the entire linker between domains 3 and 4. And I'll tell you about that linker in a second. And so you can see there's on the x-axis, this is the plot of every position in S5A. And this is the plot of every amino acid that it mutates to. The stop codons here are on the top, so you can see those are all red, which kind of makes sense. Mutation stop codon anywhere in the protein, at least in this part of the protein, is loss of function. There are these kind of interesting proline horizontal stripes, which kind of correspond to the structured alpha helices. So prolines are kind of notoriously bad for alpha helices. And there's you kind of pick up a little bit of the kind of the helix structure of the S5 helix, the S6 helix, as well as this P loop, which actually loops back into the membrane and helps form some of the pore of the proteins. That makes sense that a lot of residues in this region are loss of function. We interestingly didn't see much loss of function in the S3 and S4 helices, which I think reflects the fact that they're kind of further away from the central pore, as well as there's actually 4 of these domains, right? And so it might be that there's the protein's actually pretty insensitive to some mutation in one of the 4 domains, because it's not actually replicated in the other 3 domains. So I think that kind of illustrates some of the resilience of this protein to mutation. I told you I'd tell you a little bit about this 3,4 linker. So the 3,4 linker is actually a really interesting part of this, of voltage gated sodium channels. It forms the inactivation gate. So it forms part of the after the channel opens, it forms part of the region that loops in. And the old thought was that it directly plugs the pore. I think with the structure being revealed, it's a little more complicated than that. It kind of goes next to the pore and kind of changes the structure of the protein. But interestingly, there's some interesting gain of function residues in this assay that we discovered. Those are in blue here, and a lot of those correspond to some of these inactivation gate mutations. So maybe loss of inactivation is kind of results in gain of function, essentially in this assay. So this is previous patch clamp data from 1992, studying one of these variants, which actually opens and then just never inactivates. So it kind of just stays open, and you can kind of some of these. So we're kind of going to follow up some of these kind of interesting residues as well. This is the correlation between our did we get the right answer? So can we compare our DMS scores to patch clamp data? And we've actually patch clamped about 60 variants in this gene on the synchro patch. I'm sorry, in this region of SCN5A on the synchro patch. So we did a correlation analysis between our synchro patch peak currents on the x-axis, scaled from 0 to 100, where 100 is wild type and 0 is loss of function. And then our DMS scores kind of scaled where this is normal function and this is loss of function here. And so you can kind of see there's broadly kind of 4 quadrants. And essentially, we kind of get the right answer for a good chunk of the variants, where variants in the upper right or bottom left are kind of normal in both assays or abnormal in both assays, respectively. And obviously, there's some interesting exceptions, and we're currently investigating some of those to see if there's some, different biology, if those are an error, or if that's some interesting biology that we're revealing, and try to figure that out. So I want to expand out beyond. I wish I'd reshown the cardiac action potential slide, but to expand beyond the this SCN5A gene to other genes we're working on. So one of the other genes we're working on is a set of 2 genes, actually, KCNE1 and KCNQ1, which together encode a voltage-gated potassium channel. And that acts, if you remember the cardiac ion channel diagram, they act at the end of the action potential to help repolarize the cell at the end of the action potential. And mutations in this gene are responsible for 2 of the major forms of this long QT syndrome arrhythmia. So that's kind of why we're interested in, one of the reasons why we're interested in figuring those out. So our goal, I should say, a lot of this is led by was led by a talented MD PhD student, Aisha Muhammad. And so our goal is to kind of comprehensively characterize these 2 sets of variants to better predict these 2 types of long QT syndrome. And this is a preprint, so if you're interested in further details, you can go check it out. We tested KCNE1 function with kind of 2 different assays. So we made a library of nearly every possible. So I should say KCNQ1 is the major subunit, and KCNE1 is an accessory subunit which binds and activates KCNQ1. KCNE1 goes to the membrane once. And KCNQ1 goes to the membrane 6 times and forms a tetramer to form a full channel. So we first started with KCNE1 because it was the smallest arrhythmia-associated gene, so it was a good kind of place to work on. And so we made a library of nearly every possible variant, made 2,500 variants in this protein, 96% of all variants in the gene. And then we did 2 different assays, either a trafficking assay, whether it reached the cell surface or not, or a fitness assay to measure actual potassium channel function. So I should say that the general hypothesis we're testing here is that one of the major mechanisms of loss of function for cell surface ion channels is a failure to get to the right place. A lot of these are misfolded and then just fail to traffic to the cell surface. You can imagine other types of mutants that are loss of function because they actually get to the right place, but then they just don't work at the cell surface, or they gate improperly. But our hypothesis was that by developing these trafficking assays, we could test this major mechanism of loss of function. So this is how we do our surface trafficking assays. So what we do is we integrate our library of mutants into cells, and then we actually stain them. We have an HA tag on an extracellular bit of the protein, and we use an antibody against the HA tag in live cells so that we're only detecting the surface protein. We're not detecting any intracellular protein. And so we get some mutants that we get a lot of staining for, and then we get some mutants that we actually get little to no staining for, and those are our loss of trafficking mutants. And then on flow cytometry, we're able to sort 4 different bins, sort by level of staining, and deep sequence each bin, and essentially use those abundances in the 4 different bins to kind of infer a trafficking score for each variant. So this is what it looks like for the map of KCNE1 cell surface trafficking. So on the x-axis is position in KCNE1, and on the y-axis is every possible amino acid mutation. So you can see there's kind of lots of interesting biology going on here. I don't have time to kind of walk you through all the different interesting bits that we've noticed, but a few interesting things. So one of them is the stop codons. So stop codons early in the protein are loss of trafficking. And then at some point about halfway through the protein, you can actually chop off about half the protein and get a protein that traffics relatively normally or sometimes even gain of trafficking to the cell surface. So that's kind of an interesting observation that the C-terminal half of the protein is kind of dispensable for trafficking. There's this, I'll kind of draw your attention to two example variants. So this variant here is L51H, which is a known variant from the literature that was loss of trafficking and loss of function. So it doesn't get to the cell surface and it doesn't work. And that variant was loss of trafficking in our assay, and it was kind of an exemplar of this kind of broader group of the wild type is hydrophobic and sitting in the transmembrane region in the lipid bilayer, and then mutation to a polar or charged residue, hydrophilic residue, results in loss of trafficking. And that's kind of exemplified by this L51H residue. There's another, probably the most famous KCNE1 variant is D76N. And that variant actually had completely normal trafficking in our assay. And that makes sense because in the literature it had been described as a normal trafficking variant, yet it had defective activation of KCNQ1, had defective gating properties. There's lots of other interesting biology. There's this, just point out one more thing, the cysteine, extracellular cysteines, mutation to cysteines cause gain of trafficking. We have no idea why. We hypothesize that maybe there's some disulfide bond that's being formed with something, with maybe another KCNE1 mutant, or maybe with a different protein or something. That's a kind of an interesting future investigation, but that was kind of a surprising result. And I think kind of illustrative of the type of result that you get. You get these kind of global observations of patterns that you don't necessarily get from single variant studies. So then we spent a long time trying to develop a functional assay that not only measured whether it got to the cell surface, but whether the protein worked. And the real problem, again, for this protein is that you can express KCNQ1 and KCNE1 in a hex cell, and at the resting potential of a hex cell, there's very, very little channel activation. There's almost no flow of potassium, and so the cells are just totally fine, no fitness defect whatsoever. So what we hit upon, this is kind of a nice idea by Aisha, is to actually use a gain of function KCNQ1 variant that has a big left shift in the voltages of activation. So that's shown here. And so actually now, when you mix this S140G mutation in KCNE1 together with KCNE1, you actually get a decent amount of potassium efflux at the resting potential of a hex cell. And so what this means is that if we grow cells for 20 days expressing this is now a test of different mutants in KCNE1. For wild type, the mutants basically, wild type cells for wild type KCNE1 essentially die out over the course of 20 days, get depleted from the population, versus some loss of function variants actually stick around in the pool because there's still no efflux to the channel. So we're able to use this in the following way. So we made our pool of all the different mutants now in this cell line that's already expressing the KCNQ1 S140G mutant. We grew the cells for 20 days. Some of the cells died out, some of them didn't. And then we used high throughput sequencing of these different pools, essentially to count the frequency and kind of infer which mutants had normal function or loss of function. So this is the map that we get now for a function of KCNE1. So kind of a little different. And I should say we're not really powered in this assay to look at gain of function variants, unfortunately. So it's kind of only loss of function, only the red, none of the blue. And so essentially red is loss of function and white is kind of normal function. And so you can see again, this L51H variant is again loss of function in this assay, which makes a lot of sense. But now we actually also see this D76N variant, which has defective gating properties, also is loss of function, and indeed nearly any mutation in D76 is loss of function. And so we did a bunch of different analyses, kind of on the structure of this. There's no solved structure of KCN. There's no full like cryo-EM structure of KCNE1 in complex with KCNQ1, et cetera. But there's a kind of, we built one with AlphaFold, and we did some homology modeling with a structure with KCNE3 to try to kind of infer, this is something we think is approximately what the structure looks like, where you have KCNE1, KCNQ1 here, and then there's also calmodulin in the mix, which actually is when they solved the cryo-EM structure was kind of obligately as part of the structure. And so this is a kind of a heat map of KCNE1 only, and where the loss of function variants are in red. And this is the membrane here. So this is intracellular and this is extracellular. And you can see that in the full complex, a lot of the regions that are loss of function are actually positions that make contacts with either KCNQ1 or calmodulin in the structure. So that was kind of a cool validation of some of our scores, and maybe part of the mechanism for how you get these gating mutants. Okay, so with the last few minutes, I'll just tell you about some of the new work that we're doing on a third protein called KCNQ1. The gene's called KCNQ1. And so this is kind of the major component of this complex. And we figured once we figured out how to do the smaller gene, we'd kind of expand out to the bigger gene. So this is the gene here. It goes to the membrane 6 times and forms a tetramer to form the final protein. And so we made a barcoded library. Again, we get about, sorry, this is not updated, but we get now of the 13,000 possible variants, we get about 92%, I think, of the possible variants. So we're studying about 13,000 variants in this experiment. And so far, we've only worked out a trafficking assay. We'd love to get a function assay working, and that's kind of in progress. So here's our scores by functional class for KCNQ1. So you can see that our nonsense scores essentially are all pretty low. Our synonymous scores are mostly pretty high. A few outliers, those might be real, those might be experimental noise. We need to investigate that. And then the missense variants are kind of bimodal. So there's a lot of missense variants that traffic pretty normally to the surface. There's a lot of missense variants that are loss of trafficking. There's a few missense variants that are kind of in the middle. And there's also a little pool, if you squint, of kind of gain of trafficking variants that actually traffic a little better than wild type. And so this is the structure of KCNQ1. Goes to the membrane 6 times. There's kind of 6 main alpha helices. There's also a series of intracellular alpha helices in the C-terminal part of the protein. So here's the KCNQ1 trafficking map. So this is pretty complicated, so I'll try to kind of walk you through it. So basically on the x-axis is every position in the protein, and then the y-axis is every possible mutation. It kind of broke it into 3 lines, the first third, the second third, and the third third of the protein. One of the cool things is that a lot of the kind of major transmembrane helices kind of come out of this map. So you can see, for example, especially the S5 and S6 helices are very, very resistant to mutation to hydrophilic residues, which are this top half of the residues. And that makes a lot of sense because they're naturally hydrophobic. This P loop, which kind of loops back and forms part of the pore, is very intolerant of any mutation, essentially. And then you also see a little bit of the other helices, although again, the S0 through S1 through S4 helices are kind of less pronounced compared to the S5 and S6. There's also some other interesting biology that kind of came out of this. So one of them is this 3 amino acid motif here in the N-terminus. This is an intracellular region. The N-terminus is kind of mostly pretty tolerant of mutation, but there's this 3 amino acid region that's loss of function. Oops, sorry, this is actually out of order. So this was actually discovered in 2004, this motif. So this is a paper from 2004, a 3 amino acid LEL motif from 38 to 40 interacts with these other proteins, which help promote it to the trafficking to the cell surface. So that's kind of a nice validation that we get kind of similar results. Likewise, there's a motif near the C-terminus that actually results in gain of trafficking, which was pretty intriguing to us for a while until we read this paper from 2007. And this is a paper describing that this is actually a binding site for a E3 ubiquitin ligase. So this is the motif that the E3 ubiquitin ligase recognizes, the so-called PY motif. And that results in the ubiquitination and kind of degradation of partial degradation of KCNQ1. And so we think that mutation of this motif would cause increased kind of steady state abundance of the protein, which kind of makes sense for these increased trafficking scores. So I think that's really cool that you get some of this known biology recapitulated in this map. There's also some stuff that, as far as I can tell, there might be a paper from 2006 describing this, but as far as I can tell, is relatively novel. So this N-terminal region, for example, there's this really interesting pattern where mutations to charged residues affects trafficking. So mutations to negative charge results in increased trafficking, and mutations to positively charged residues results in decreased trafficking. We don't really understand this yet. If anybody has any ideas, I'd love to hear them. Or if anybody was an author on the paper from 2007 showing this, I'd be love to hear it. We're still kind of staring at a lot of these, and obviously I'm not really doing justice. You can spend 3 hours investigating each of these residues in the middle of the protein and try to figure out why it's working and why this one's red and why this one is white and so forth. But there's kind of isn't enough hours in the day to do all that. One of the analyses we have done is we've looked at the correlation. Let me back up for a second. This is the correlation with 51 previous manual trafficking measurements, one at a time. So we get really nice correlation with the manual trafficking measurements. And then this is the correlation with known disease risk. So essentially, we're getting normal trafficking for almost all of the described benign variants. So that's really great. We're getting abnormal trafficking for about 2/3 of the described pathogenic variants, and normal trafficking for about 1/3 of the described pathogenic variants. So I think what this is illustrating is essentially that about 2/3 of the pathogenic variants are acting through a loss of trafficking mechanism, and about 1/3 of the trafficking variants don't act through a trafficking mechanism and instead act through a gating mechanism. So there's some famous gating variants that are up in here, for example, that have normal trafficking in our assay. So we'd really love to get a functional assay working, and we're kind of working hard on that. So kind of overall, I'll just sum up and say that, I'm really optimistic that deep mutational scans, this kind of comprehensive scanning approach, can provide insight into disease risk, but also into kind of basic protein biology. And so we can kind of help identify important trafficking and functional domains. And then ultimately, if we prove that these assays are good enough, the hope is that we can kind of integrate these assays into the classification scheme to help actually help address this VUS problems. When a new variant shows up in a patient, we've actually pre-calculated the scores in our demutational scan and kind of use that data. I'll mention a couple of major limitations. We're using a non-cardiac cellular system without the full complement of cofactor genes. We're simplifying the complicated behavior of a channel to a single number. We're not modeling all the complicated regulation and properties of these channels. And so I like to think of this as kind of a pyramid scheme, which I know has a negative connotation, but I think it's kind of a nice analogy for what we're trying to do is, obviously there's lots of groups, including us for a couple of variants. We're trying to do detailed kind of lower throughput variant studies. We're really solving how single variants or a few variants are working. And that's, I do not want to replace those studies. I think those studies are really important to figure out kind of new mechanisms and phenomena. And instead, what we want to do is kind of supplement those with kind of big tiers of automated patch clamping or deputational scans, which can kind of study hundreds or even thousands of variants and kind of broaden those out to all of the different rest of the variants in these genes. So just a couple acknowledgements. A lot of this work I started when I was a postdoc in Dan Roden's lab, and he's been kind of a close collaborator even in my new lab. So that's been a really great collaboration. There's been lots of people who did a lot of these work and funding. And if you're interested in, we're interested in expanding these out, even beyond the cardiac world into other types of ion channels. So if you're interested in these types of high throughput studies, as a postdoc or potential collaborator, feel free to get in touch. And thanks a lot. And I can take any questions.
Great talk, Andrew. Thank you very much. We've got a question in the chat from Jian Gao, who says, good talk. I wonder, do cell densities, medium type, or other conditions affect the result of the cell survival method for the deep mutational scanning?
Can you say it one more time?
Whether you make considerations for cell density or the media that the cells are bathed in, how much have you explored how that can change the survival phenotype?
Yeah, I think that's really important. One of the things we've noticed is that hex cells attach to each other. So the cells actually have to be pretty sparse when we add the drugs, so that each cell kind of makes an independent decision whether it dies and floats off, versus if they're in a lawn, you get a lawn of cells that float away. And yeah, definitely we've played a lot with the different media and different drug concentrations and different timings, and that stuff all probably does matter a lot. Obviously, like ion concentrations probably matter for these ion channels and things like that. So those are definitely parameters to kind of tweak as you develop these assays.
A second question. Great talk. This is from Michael Netzer. Do you have any plans on working on CACN A1C variants?
Yeah, that's a great idea. Yeah, I think we have some kind of vague plans to do that. Where we have we have a big grant that got funded together with Dan, as well as Brett Cronky, who's another early faculty here, and myself, to study lots of different cardiac ion channel genes, demutational scans. So that's one of the ones that was on our list. So we haven't really done too much on it yet. It's again, yeah, CACNA1C has the same challenge, which is that it's also 2,000 amino acids long, and also has lots of complicated regulation, and also has multiple other cofactor genes. So it's going to be a complicated one to do, but yeah, we would love to do that one in the future.
David Colhoun has a question. Greg, maybe can you elevate him to a panelist, please?
Sure.
In the meantime, maybe I'll ask one, Andrew. So in your patch clamp data, you not only get the peak current, of course, but you can get all sorts of other rich functional data, activation voltages, inactivation half potentials, recovery from inactivation. Have you mapped any of those effects of mutations to see whether you can pick out any other mechanisms by which you arrive at these diseases?
Yeah, the one idea that we've tried on that front is to try to deliberately make hobbled versions of the protein which lack certain features. So for example, we have this mutant that just has no inactivation. The inactivation of the channel is gone. And so we made the whole library in that background. And the idea is if we get scores for that pool of variants, then we can kind of infer, by comparing the scores in the normal context versus comparing the scores in the inactivation deficient context, we can kind of start teasing apart the different features of the protein. Right. I don't think we're ever going to have the resolution where we're going to be able to say, this channel has a 6.3 millivolt shift in the voltage of activation as judged by the deep mutational scanning. Right. But yeah, I'm hopeful that we can start to kind of one of the principles is that once you make this library and once, it takes years to kind of make all the infrastructure and get the scan working, but it takes only maybe a month or two to repeat the scan in a slightly different context. So in a context with also expressing another cofactor, with under different, temperature or drug condition or something like that. So I think that's one avenue that maybe kind of repeated scans of the same gene in slightly different altered contexts, I think would be really exciting.
Yeah, maybe drug screening for personalized medicine effects. Yeah. Anyway, right. I'll pass on to David.
Yes, thank you very much. It was a very interesting talk. As a person who spends about a year on each mutant, or used to when I was active, that's it's quite a revelation. I've got two questions though. One is this first part of the talk, the sodium channel one, didn't seem to distinguish between trafficking and an altered function at all. That was the second part of the talk. But could some of these sodium channel mutants be decreased trafficking rather than?
Yeah. So we've tried. So every other gene that we've tried, we can pretty easily make a small epitope tag in an extracellular region that we can use for these trafficking studies. We tried really hard to get one for SCN5A, and we just couldn't get it. We tried all the ones that were published in literature. We tried a few ourselves, including guided by the structure, and none of them seemed to work. And so we don't really have a really efficient trafficking assay for SCN5A, but I would guess that a lot of the SCN5A loss of function variants would be acting through a trafficking mechanism, and I wish we could tease that apart. We have done some, kind of lower throughput Western blot measurements of trafficking and things like that, but we haven't done it at scale.
Good, thank you. And the second question was concerning your more detailed studies. I'm glad you're thinking of doing some of them because I find them very interesting, at least. But they've never actually been done successfully, to my knowledge, with a voltage-activated channel. It's all ligand-gated channels. I think perhaps there's two reasons for that. One is that with concentration, you've got a proper zero. With voltage, you haven't. It's minus infinity, I guess, the zero equivalent of zero concentration. And the other one is, I forgot what the other one is now. I'm getting too old for this. Anyway, that's one reason. You know, with the glycine receptor, we can fit 14 free rate constants to 10 different states. But that nothing of that sort has ever been attempted with a voltage activated channel, and I think there's probably reasons for that.
Yeah, I think part of it might depend on what your definition of detailed is. That might be a little beyond my area of expertise in terms of how you would go about doing that level of detail for the modeling of the different states and things like that. And you know, certainly there's other people who are working on lots of really interesting modeling efforts, you know, both on the structural level as well as kind of the different states and things like that. And I'm probably not the best person to ask about that.
Yes, I suspect. I mean, I started off with great glee, thinking we could solve the structure-activity relationships. If only we had the structure of the receptors. Then we got the structure of the receptors, and I still thought this is going to solve it all. And it's really hardly further ahead than it was 50 years ago, I don't think. I suspect that one reason could be is that forces that vary as r to the sixth are going to vary enormously when separations of things change to an extent which is not even perceptible in the best X-ray crystallography. It's all very, very complicated.
Yeah, well, I would love it if, you know, I'm trained as a geneticist, so this is definitely outside my comfort area, but I would love it if some of these high throughput data sets, some bright mind could use them to help discover something new and make some new simulation of how these proteins are working.
Thank you very much.
One last quick question in the Q&A, Andrew, from Xiping Pang. Great talk, very powerful and very interesting. A technical question for the synchro patch system. Do you do series resistance compensation?
Yeah, we can. Yeah, we typically do. Yeah.
Okay, so I think that wraps up all our questions, and we're 5 minutes over. So we'll say thank you very much, Andrew. Thank you for joining the series. Another fantastic talk, and we will see everyone next month for the next seminar, which is on the 3rd of October, and will be Michael, Martin, Aragon, Bardell, discussing vascular calcium channels. So we look forward to that. So thanks a lot, Andrew. Hopefully see everyone next month.